I'm glad you can all join us. Today, we'll be having a chance to hear from 3 of the QuestCap partner companies. We'll be here from Glenco Medical Corporation, Dr. Glenn Copeland. We'll hear from Amino Therapeutics, and we're going to hear from Spectral Analytics. Each of the companies will be given a chance to take a few minutes, explain what it is they're doing, to give everyone an update on exactly the exciting initiatives that we have going in each of these companies. Before we start, I'm going to talk a little bit about getting Aaron, our legal attorney, Aaron Aiton. He is going to read a disclaimer. Aaron, are you available?
Yes, happy to.
Okay, why don't you go ahead and do that?
All right. The purpose of this call is to give QuestCap's partners the opportunity to provide an update on their work. Select representatives from the MedQuest division will also make short representations on their progress and answer questions from shareholders. The views, information, or opinions expressed by our partners are solely those of the individual presenters and do not necessarily represent the views of QuestCap and/or its directors, officers, or employees. QuestCap is not responsible for and has not in all cases verified for accuracy any of the information to be provided by our partners on this call. Neither QuestCap nor its partners are making any express or implied claims that any of the research or products discussed on today's call have the ability to eliminate, cure, or contain the COVID-19 or SARS-2 coronavirus at this time. During this call, QuestCap may make statements that contain forward-looking information within the meaning of applicable Canadian securities legislation. Forward-looking information will include, but is not limited to, statements with respect to the potential of any of QuestCap's assets and investments, the ability of QuestCap to monetize on any of its investments, the pursuit by QuestCap of future investment opportunities, and the future financial and operating circumstances of QuestCap and its assets and investments. Forward-looking information is subject to known and unknown risks, uncertainties, and other factors that may cause actual results to be materially different from the conclusion, forecasts, or projections expressed or implied by such forward-looking information. Certain material factors and assumptions have been applied in drawing the conclusions or making forecasts or projections reflected in the forward-looking information. A description of the factors that could affect and the assumptions underlying forward-looking information can be found in QuestCap's annual information form, financial statements, and accompanying management discussion and analysis, as well as public disclosure documents that are available for your review under QuestCap's profile on SEDAR. Participants should not place undue reliance on forward-looking information. QuestCap does not undertake to update any forward-looking information except in accordance with applicable laws. And that is the end of the statement.
Great, Aaron. Thank you very much.
Thank you.
What I wanted to do before we turn it over to Dr. Copeland at Glenco Medical, I wanted to talk a little bit about some of the things we've been doing, leading with Glenco Medical as the face of a lot of the programs that we do for a number of companies within QuestCap. We're really talking about programs and not about products. What we want to do is really lead with various types of standards to get people back to work, get people back to play, get people back to school. These discussions and these intros from Glenco have actually led to discussions in North America, in South America, in Europe, and in Asia, with a lot of very large corporations, large institutions, about ways we can help them with the protocols to get them back to whatever it is they need to do and get them back there safely. One of the key components of this, of course, is testing. And I did want to update everyone on the testing side. We were disappointed when PCL, the company that we had a distribution agreement with through the MTGR group, were rejected by the FDA for the emergency use authorization. They weren't alone. 94 serology tests have been rejected as of this morning by the FDA. 11 have been accepted. We were getting concerned with the length of time and the number of rejections that were coming from the FDA. So we began to look for an alternative that could serve us well within the US and the North American marketplace. We were able to acquire distribution rights for the Lear test, and the test is an EUA approved product, and it actually is one that we have now begun to bring into the United States to fill some of the customer demand that we've been able to acquire. Testing is going to be extremely important, and more so in the near future than it is even right now. Currently in the US, they test about 5 million people a week. the Rockefeller Foundation recently published a report that indicated come this fall, when the cold and the flu season starts, the need to discriminate between COVID, cold, flu will increase that demand to probably closer to 30 million tests a week. That testing is almost an unsatiable demand that we're going to see. And as a company, we're looking at programs, initiatives across all of the different testing modalities, but more importantly, across all the different programs that we would need to really be able to safely bring people back to a normal way of life. So that is what we're focusing our efforts on now. We will be announcing some very interesting and exciting initiatives in the very near future. And with that, I wanted to take the opportunity now to turn things over to Dr. Glenn Copeland with the Glenco Medical Corporation. Dr. Copeland?
Doug, thank you. Just to carry on with exactly your theme, Glenco has taken a lead role with QuestCap to try and determine the direction of testing. You know, this virus right now is approximately 30 weeks old in North America. So it's been, if you will, a brand new challenge for humanity. It's been a brand new challenge for medicine. It's been a brand new challenge for physicians who have to deal with this on a day-to-day basis. One of the early areas that we got involved with QuestCap was the first one was return to sports, and we were put into a great position of working with all the professional leagues and looking at the way we could return to safe sports. In fact, our first protocol was actually called Return to Safe Sport, which was reviewed by all the major leagues. And we were the first ones, I'm proud to say, to talk about a hub, which was, as we referred to it, as the inner circle, which right now has proved so far to be very successful in the NBA and the NHL. We looked at it with one of our great consultants, Dr. Larry Steinman, who is professor and chairman of pediatric at Stanford and an immunologist. Larry and myself and my team at Glenco worked very, very long hours to put together these protocols. As time went on, Doug, what we did was look at all the different testing. So we had the ability early on to work with the PCL rapid flow, lateral rapid flow antibody test, which showed us the availability of isolating two different antibodies, IgM and IgG. However, COVID testing has dramatically changed over the past 26 weeks. Today, when we started, there was a PCR test, a nasal swab test. Unfortunately, the demand was so high early on that results 7 to 10 days to get, which unfortunately is not a great thing in the medical world. Physicians treating this disease need to know results much faster. So QuestCap and Glenco took a very, very strong stand at looking around the world for leading tests, whether it's been rapid antigen tests, whether it's been a rapid PCR test, rapid antibody testing. We've isolated all the great manufacturers around the world. And where we're at right now is leading once again in determining what are the best tests to use in each given situation. So for argument's sake, generally speaking, a PCR test will isolate the virus, but usually they take at least 2 to 3 days, whereas a rapid antigen test would give us a readout using blood to find out whether there is an antigen present. And an antibody test, rapid, can tell us whether you have antibodies, you've been exposed, or with IgM, whether you have been very recently exposed and are starting to show signs of contracting COVID. So we're working very closely with a great team of our medical advisors to put together the, as I like to refer to it, as the standard of proper testing. And this standard sets about, which test is appropriate. What would you use for a professional team? What would you use for teachers going back to the classroom? What would we use for people going back to office buildings? What do we use for bus drivers? So everybody needs would be different. And how often do they get tested, and at what level? So we're very excited about, once again, leading in the prevention of the illness, and most important, the spread of the illness. That's been our real focus. How do we quickly isolate a person who's been shown with the disease? How do we quarantine them? How do we make sure people around them didn't get sick or are sick who need to be quarantined? The key is to stop the spread. In Canada, we have done an outstanding job in preventing the spread. The numbers are dropping. I think we're one of the best countries in the world for the great work that our public health team has done, and our infectious disease docs, immunologists, epidemiologists, and I'm hoping we can help this world open up. So with that, Doug, I'll turn it back to you, and look forward to reporting at the next webinar on the great successes we're having.
Yeah, I mean, one of the things, Glenn, just building on some of what you said, I mean, the programs that you're initiating are more than simply testing. You indicate also temperature checking. You have survey, or you have symptom questionnaires. You really want to understand more than simply the results of the test. It really is understanding the patient, the entire environment around the patient.
Not only that, Doug, but the data collection we're doing, and being able to grab this data and start looking at epidemiology studies, if you will, because we're taking all the data from well people and finding out how long they stay well, when and if they get sick, what happened, how did they get the mode of transportation of the disease. And we're looking at all these different ways of, again, always zeroing in on stopping the spread. So we're changing almost weekly, Doug, and it's exciting to be part of the QuestCap company.
Dr. Copeland, the other thing I think, just for sort of education of the folks, and I'm the one getting educated from you as well, but we have really, you mentioned 3 types of testing. You mentioned the PCR test, which is one most of us are familiar with on the news, where you see someone pulling up to either a drive-through or having a nasal swab put down their nose to get a result. The standard for determining, I believe, whether or not someone is actually contracted the virus.
Right.
That right now is what most people are using. One of the things that comes out very clearly in the Rockefeller report is that doing 5 million a week right now has completely jammed the capacity of the labs in the US, because it's all still to be done through high complexity laboratories in the United States. There are horror stories of people waiting 2 to 3 weeks to get the result back, when they could either be walking around positive or have been negative at the time of testing and now have become positive in that 2 to 3 week period. So it's certainly not acceptable. Your programs address all components of that, address the PCR, address also the capability of the antigen, which is also a test, I believe, that can determine if the virus is present or not. But perhaps a day or so later when the antigen forms. So it's not considered quite the standard of the PCR, but a great ability to take care of some of this capacity.
Right, it's an adjunct test, Doug, and you're exactly right. It'd be great to have available PCR rapid testing. So you do your test, and that night, or maybe within 24 hours, we get the results. But the problem is that the medical laboratories with their machinery can't do it that quickly. It's just right now impossible. And as you said, some people are waiting 7, 8, 10 days, 12 days. And that, that's just unacceptable. I mean, think about doing a strep test. Everybody knows, and finding out, 10 days later that you had strep. We need it now. We need to know, what's happening to our bodies now. And that's why when we say the rapid testing usually gives results within 4 hours. So although an antigen test usually takes 2 or 3 days for the antigen to be positive in the blood, you get a readout very quickly. You couldn't have a PCR test today and be waiting 5 days, where in 2 days you have an antigen rapid test and you'll have the result long before you get your PCR result. So we're trying to modify the testing and look at the different tests available to ensure that the population gets the quickest results possible, so they can get into quarantine, get medical care, do the appropriate things. And I think QuestCap Glenco is leading that charge.
The other thing I think that you make a strong recommendation on to every one of the programs that I've heard you present to is the need for antibody testing in all cases to supplement the antigen or the PCR. They're simply telling you whether or not the person is actually carrying the virus. The antibody will tell you the status of the antibodies that have formed. And I guess importantly, the IgG, which is potentially the immunity antibody, and can not only indicate there may be that immunity, but begin to monitor the durability of that, how long that lasts.
Exactly. Depending on what you read in the literature, and I'm talking, well-documented literature, we suggest that there could be anywhere between 4 and 20 times more people who have been infected, but may have stayed asymptomatic. So, if those numbers are correct, and, we're just looking at the literature, that means for every one person that has been proven that they had COVID, there's could be anywhere between 4 and 20 times more. So the IgG testing, the antibody testing, we're finding that people are coming in and you do the antibody test, and these antibody tests, by the way, Doug, are all FDA, EUA approved, and QuestCap again has led the charge on this. But these people have IgG, which means they've been exposed and they've developed viable antibodies. So the problem is that you have some immunity. We don't know exactly for how long. Listening to Dr. Steinman, who is an immunologist, he believes that it could be anywhere from 6 months to who knows. But the great part is that if you are IgG positive, meaning that you've been exposed and you've developed antibodies, it doesn't give you a free pass. It doesn't say that, oh, I don't have to worry about this. But it gives us an opportunity to follow that patient and see how long the antibodies are in the blood, how long they last for, and how long they might be implied immunity, which is an exciting project that Glenco is leading.
We had an opportunity to talk to the head of the Canadian Immunology Task Force, the task force set up by the Prime Minister to look at immunology in Canada. We had Professor Steinman on the call, and I think certainly they recognize, if not at the beginning of the call, certainly by the end of the call, the absolute requirement for understanding the antibody status of the patient population in Canada. So yeah, I think there's some exciting times ahead around testing overall, but specifically around the antibody. There's also tremendous demand we're seeing for people who think they may have been exposed in the past or had a cold back in January. There seems to be almost an insatiable demand for people wanting to get tested for antibodies to confirm whether or not they were exposed. So I think exactly. I think all of those testing methodologies are going to be not only in demand now, but certainly, as I mentioned earlier, this fall they're going to become very much in demand. From some of the reports you read about the need to differentiate between colds and flu and COVID, just for basic treatment options on the different patients.
That's going to be a major issue for us, Doug, especially in the northern part of the hemisphere. We're coming within, say, 10 weeks. We're going to be into cold season, flu season. and unfortunately COVID season. And how do you differentiate between the three? And it's going to be very important for family doctors and all medical people to be able to get rapid results, to know what you're dealing with. Because obviously COVID is treated differently than seasonal flu would be treated versus a sinus infection that you might get with a simple cold. So testing is going to become a major contributor to the right treatment at the right time.
Great. Well, Dr. Copeland, thank you very much. At QuestCap, we're continuing to investigate all the different, you made all the different testing methodologies. You made the comment rapid, and that is really what we are ensuring we are focused on, because waiting days or weeks for testing is not going to be acceptable. We want to be able to also take the testing within the regulations as close to and conveniently to the population. And we've got some interesting initiatives that are going to be announced in the very near future about what we're doing on that side. So Dr. Copeland, thank you very much.
Thank you, Doug.
You're welcome. I'd like to turn it over now to David at Amino Therapeutics. David, the floor is yours, sir.
Great. Well, thank you so much, Doug, and appreciate everyone joining the call today. And that was a great, thank you, Dr. Copeland, for sharing. That was a great update on your project. Here at Amino, what we've been doing is basically focusing on how we can design peptide-based protease inhibitors that would ideally halt the function of the main protease in SARS-CoV-2. Interestingly enough, we weren't the only group that thought this was a good idea. Shortly after we made an announcement with QuestCap to pursue this endeavor, we saw a published paper in Nature the following month with successful results. So we were actually, we know it's a good idea. There are 4 main binding cavities of 140 known ligands for the main protease. So there are a lot of targets that we can go after. So it doesn't necessarily have to overlap, 100% with one of the competing groups out there. It's from academia, so it's not exactly a competitor. But basically, what we have is a lot of computational modeling done to identify, essentially a way to take a protease which can use and incorporate essentially components of other viruses that are known to penetrate the lipid bilayer of a cell membrane, and essentially have that on one end of the protease. And all the protease is a small, protein or peptide, essentially. And essentially, so the idea is to have a compound that will cross the cell membrane and then inhibit the function of the main protease, just using different combinations of short amino acid sequences. That's basically it. What we've done to work towards that progress, in addition to some computational work, the design process is to establish a 5,000 square foot laboratory space in a certified GMP manufacturing facility that's in Ashland, Mass, so about 45 minutes west of Boston. We've got maybe 2/3 of the equipment we'll need to kind of commence operations there. So we've seen, good progress, and, we're just looking forward to kind of the next steps. Essentially what our process will be is to finalize the computational models, synthesize the peptides in vitro, and we'll probably use a third-party testing facility, maybe one of the big research hospitals here in Boston, to facilitate the actual assays against type 2 immunocytes that are infected with SARS-CoV-2.
David, from a commercial standpoint, can you share with everyone sort of what the commercial plan is once the research reaches that critical stage?
Absolutely, yeah, it's very easy. All we would do is take the data and bring it to a large pharma company. Every day, almost daily, we're seeing deals where giant pharma is buying up or doing, multi-million or even multi-billion-dollar deals with, much smaller pharmaceutical startups, and it's primarily because that's a more cost-effective way to do the upfront research. So all our plan is to try that initial assay, and if we're successfully able to inhibit viral replication in human cells, then that's it. All the animal studies and future results would come from a large pharma company.
Okay, I mean, I personally have some experience with that. I was for many years with a company called Teva. We were both brand and generic. We tended to not want to start from scratch. We let the smaller companies do that development work. We would actually enter after a phase 2 in many cases. There's a current migraine drug on the market for Teva called Ajovy, and it was purchased after a phase 2. We could have probably purchased it after phase 1 for $75 million. We purchased it after phase 2 for $435 million. It certainly de-risks the project, but it does certainly escalate the value of the research if you can pass all the critical stages.
Absolutely. And I think it's a great way to target not only, SARS-CoV-2, but in terms of the concept of cell-penetrating peptides. Approximately 60% of intracellular drug targets are intractable right now because we don't have the mechanism to cross the cell lipid bilayer effectively for biologic drugs. So anything that's a small molecule target, we can go after. But the problem is small molecules don't have the specificity that biologics do. And what's great about our approach is this sweet spot could be, a really good approach. It kind of combines a hybrid of the two approaches. You're able to go after the intracellular targets and make them, potentially druggable, which opens a whole range of possibilities. And you're doing so with a biologic, which means you can have a much higher level of specificity and to do a lot more things from a drug design standpoint that's just impossible with small molecules.
Any idea you can share on general timeframes on what your sort of work plan is going forward?
Yeah, absolutely. I mean, I think, it's taking a little longer to kind of do that upfront, side of just kind of getting established at our baseline. I would hopefully we would have results, sometime this fall. Obviously, that's dependent on capital resources, when we can get into, a lab with one of the partner hospitals. But as soon as those resources are available, we're standing by and ready to go. What we've been doing in the meantime is optimizing from a computational standpoint, essentially the best protease design. So essentially the sequence of amino acids that we're going to use to, potentially bind to any one of these 140 known ligands, or, there are really 4 or 5 main targets, and we've got kind of one that we like the best. So that's what we've been doing, is narrowing down that pipeline internally from a computational standpoint. So that way when we go to the lab, it's, very straightforward, and we can perform our experiment, make the peptides we're looking to make, and then we can assay them. And we're looking at different parallel assay methods so that we can streamline those processes and get results that, potentially a big pharma would like if they're positive, sooner than later.
Okay, great. David, thank you very much. With that, I'll now turn it over to Stephen Adler. Stephen, I'll give you the floor from Spectral Analytics.
I appreciate it. Doug. I'm actually going to turn it over to Christina, who's heading up all of our research and exactly where we are. I'm working with the FDA and the studies. So I just want to have Christina share your screen, and we can walk through exactly where we are.
Sure, no problem. Give me 1 second, Stephen. Okay. Stephen, would you like me to start, or shall you?
No, you can walk through it.
Okay, perfect. So this is just a brief agenda that we put together regarding where we are with our COVID study that we have obviously partnered with QuestCap. I am the CEO of a clinical research organization called Affinity Biopartners that's leading the operation initiative here on this clinical trial. So this is just a brief overview of who we are. So here's the deal. We have 2 FDA repurposed drugs that we're utilizing, amlodipine and mifepristone, in combination of each other. Both of these drugs have been treated with respect to Ebola and HIV patients previously. We are looking to go into 20 patients, which would be a phase 1 study for the first initial round of the clinical trial. We have identified our investigator, who is located in Columbus, Georgia. These are going to be outpatient subjects that we will be treating. This is a treatment protocol for COVID-19. The subjects will have 4 visits on site, so total of 4 days that they will be on study drug. We've built in an extra day just in case that we are not dropping the viral load as quickly as we had originally planned. And it's considered an unscheduled visit and up to the discretion of the principal investigator. Day 30 is when we have a telemed visit with these patients, where we just do a check-in to make sure that they're still feeling well, they're non-symptomatic, there aren't any issues, obviously. We'll have our full data set 1 week thereafter. So 1 week after day 30, we'll have our full data set on our 20 patients, and at which time we will be submitting a protocol amendment to increase to a 100 to 200 subject study, which then will turn it into a phase 1/2. Okay, so the study status. We have IRB approval from a central IRB. We had positive in vitro studies. We did 2 of them on infected.
Christina, sorry, could you advance your slide just so that we get the bigger shot? Or maybe start the slideshow if that's possible.
Okay, hold on 1 second, I'm sorry.
There you go.
It's working now?
That's good, yep.
Okay, good, because it looks different on my side. Thank you. So sorry, guys. So study status and where we are today. We have IRB approval, as I mentioned. Central IRB approved us pretty much immediately, within 24 hours, actually. We have positive in vitro studies. We performed 2 in vitro studies with 2 different vendors and infected COVID cells, and it was effective in the sense that it dropped the viral load, which is what we're looking to do. The FDA is reviewing our positive in vitro data. We submitted another briefing document to the FDA on 7/27 for review. The clinical trial site obviously is ready to go once we receive the FDA approval. We are working with the White House to try to expedite our FDA approval. As you noticed, it's been there since 7/27, so the CAPS is not being followed on our protocol. So we've been working with the White House daily. What I mean by that is Stephen luckily has very, very high up connections within national security, and they are debriefing the White House literally daily on where we are with this protocol. We have worldwide distribution already worked out with our generic manufacturer to be able to distribute both drugs worldwide. And we have a potential to run this clinical study in another country. Future COVID-19 studies with Spectral. We're in discussions with other potential related partners with respect to COVID studies. We have the patented cortisol reduction formula. We actually have a submission in right now under the CARES Act in Pennsylvania for a grant to do a study for this cortisol reduction with a local lab here in Pennsylvania. We also have gold nanoparticles with ribose using an inhaler. And this inhaler has already been approved by the FDA. So it would just be adding the gold nanoparticles in there. We also have cannabinoids using an inhaler device. And we have our patented peptide to reduce inflammatory load. And that's it.
Great. Stephen, just again, highlighting the commercial plan. The successful results from the patient study, there's initially, I believe, Christina, going to be 20 patients
20,
in the
correct.
study.
Yes.
And then moving to a larger study, I believe the number is 80. Is that still the correct number approximately?
Well, yes, it would be 20 plus 80, which would equate to 100 or potentially 200, depending upon what the FDA comes back to us on.
I've got it. And timing-wise on that and the potential for any sort of discussion or potential with commercial partner.
Oh, we based on our initial discussions with the two pharmaceutical companies who are manufacturing the compounds, they said after a successful 20-patient study, they would immediately go into basically a licensing arrangement on the research protocol. So basically, because they're saying 20 patients would be enough to go into negotiations, figuring it's going to take a couple of weeks while we ramp up the number of patients from that 20 to either 100 or 200. So right after the successful 20, we wouldn't even wait for the safety study because, as I said, there's already a huge amount of data on the safety of these two drugs. So they would immediately go into discussions on licensing.
We should also, oh, I'm sorry. One last thing too is that one thing we didn't mention is this, this combination treatment has been patented. Stephen has filed multitudes of patents worldwide for this combination to protect us.
Okay, great. And the dosage sizes, Christina, these are dosage sizes that have been approved with previously by the FDA for these particular molecules.
So yes, the FDA has approved because we are using an increased dosage for these protocols. So far, we have not received any adverse feedback from the FDA challenging why our upward limits is 3 times the normal limit. The IRB issued the approval without any questions regarding dosing. Obviously, one of these drugs has a REMS attached to it, but it's to our advantage actually, because that drug will not allow clotting to happen because it causes bleeding.
Okay. Okay, good. Anything else, Stephen or Christina, that you'd like to add for the folks on the webinar?
No, the only other thing is we're going to find out this week about potentially having the study done in another country, which again would be done under FDA guidance. But basically in advance of the FDA approval, there are other countries who might be much more interested in moving this forward much quicker because many of the other countries are not looking at the vaccine as their eventual goal. They would rather have a treatment now and the vaccine whenever it comes out rather than wait for the vaccine.
Okay, great. So what we've heard from today are 3 very different type of projects within the QuestCap group of companies. The Glenco model is a commercial model that is both service related and also does have the ability in a large way to drive initiation of the various testing methodologies that we have available. It's been a great entree into many very large discussions, and you'll hear about some of those in the very near future. We also have 2 treatment type of methodologies that will both be taken to commercial success under a licensing type arrangement, but both exciting, both at interesting stages. All of the information to date looks promising. So we wish everybody lots of luck, all 3 companies. And thank you for being on the webinar today. Thank you for being a part of the QuestCap family of companies. And we'll make sure we keep everybody abreast of some of the successes that you have in the very near future and continue the communication. Nick, are there any specific questions we have that I can see if anyone on the phone or myself can address?
Yes, there's some questions about test kits, the private placement, and just a little background on antibodies.
Okay, question for Dr. Copeland. Does Glenco make sure of the testing kits as part of rolling out the standard? If so, are they the QSC kits? Dr. Copeland, are you there?
I'm here. Yep. Sorry.
So as part of the standard, the testing kits, basically you as part of rolling out the standard, and again, I will try to answer this. If I'm not correct, you can certainly correct me. You put a protocol out that recommends the different types of testing for the different type of application.
Right.
As we've said, the concept is the same. Often the logistics dictate the type of testing and what's required. But these are tests that are associated wherever possible with QuestCap, correct?
Exactly right.
Okay. There was a question about, and I will address that now, about our private placement, which is closing this week. It should be literally the paperwork is being drawn up, and it will be closing in the next day or so. Certainly by the end of this week, it will be closed from a standpoint of the private placement. We have a question on, does an antibody test take longer to show up since the body takes some time to develop the antibodies? And that's a very good question, and it is exactly why we are very much, as part of the protocol, suggesting you have to have a different type of testing procedures. The antibodies will not necessarily pick up a COVID virus in the first few days of infection because the antibodies may not have formed in the body. That's why the protocols that Dr. Copeland and Glenco recommend typically start with a confirmation of whether or not the person has the virus with either a PCR or an antibody, the preference being rapid wherever possible and on site where it is allowed within regulation. And then following up with an important antibody test to not only confirm the results of the antigen or the PCR, because it will indicate an IgM or an IgG antibody indicating a confirmation of the test, or if it shows up and the test has showed negative, well then test again because it could be indicating an issue. And more importantly, also understanding the level of antibody in the population. And I think that is going to be a critical element going forward, is understanding. So most of the protocols recommend a combination of an initial test to indicate is there a virus, and then a supplemental test and an ongoing test of the antibodies to really see how they survive. So the antibody testing is the more longer term continuing testing in a number of the programs that have been established. Dr. Copeland, anything to add?
No, I think you got the nail right on the head, Doug. The antibody test is not used as an initial diagnostic test like a PCR would be used, but it would absolutely be used to screen very quickly. You mentioned the IgM part of that test can take anywhere between 3 to 5 days from the time you've been exposed to the virus for IgM to go positive. IgG is, if you will, a much later forming antibody, usually somewhere between 7 and 14 days before somebody would be IgG positive. But for all those asymptomatic people who never had any symptoms, and then to do that particular test and find out that you have antibodies gives you some sense that you've been exposed and you act accordingly. But yeah, so we're looking at each test, and an antibody test is, generally speaking, not a primary test that you would use front line like a PCR test.
Okay, great. Well, timing wise, we are 45 minutes. If there's other questions, we will attempt to get answers, and certainly we'll include answers as part of the next webinar that we hold. But with that, I'd like to thank all of our participants. I'd like to thank the guests who have joined and listened through the presentations. And we will be holding another one of these in probably a similar type of 4 to 6 week time frame to bring you up to speed with some of the additional initiatives that are underway, and you'll be heard from very shortly. So thanks everyone, and we appreciate your participation. Thank you.
Thanks everyone.